Neuromuscular disorders include a wide range of diseases, with hundreds of genes implicated in the pathogenesis. Right now, our key priorities are ensuring there are treatment options to improve symptoms, halting disease progression, and enhancing quality of life. What are the challenges that we are facing? How can we work together to better understand the genetic causes of neuromuscular disorders and drive technological innovation to benefit patients? Please welcome Karsten Baumann, Chief, Neuromuscular and Neurogenetic Disorders of Childhood Section, Senior Investigator and Acting Chief, Neurogenetics Branch, NDS, NIH. Matthew Harms, Associate Professor of Neurology, Institute for Genomic Medicine, Columbia University, and Sharon Hester Lee, Chief Research Officer, Muscular Dystrophy Association.
Well, so thank you everyone for joining us on this series on rare disease. And specifically, we're going to talk about moving the needle in ALS and other neuromuscular disorders. So the talks that you will hear later will be focused in on ALS, but we're starting right now from a very broad, big picture look at neuromuscular disease as a whole and how ALS and SMA and Duchenne muscular dystrophy and myasthenia gravis and all this broad array of diseases actually fit together in a category. And I think if you think about neuromuscular disease now and the huge breadth of them, it may not seem like they all have a lot in common, but if you put yourselves in the place of physicians a couple of 100 years ago, we're seeing patients, and the one thing that these patients have in common usually is muscle weakness, and it's often progressive muscle weakness. And so the group of physicians studying these diseases tend to see all of these people together, and they really didn't know if this was an acquired disease or a genetic disease, if it was neurological in origin or a muscle disease, or even secondary to something completely different. And where we have sort of fallen out in the field is that organizations like the Muscular Dystrophy Association or the AFM in France, TRINMD in Europe, cover this broad range of neuromuscular diseases that include diseases that cause muscle weakness either because of primary underlying muscle problems or something related to the nervous system that controls voluntary muscle movement, or that could be central or peripheral. And so you end up with this broad group of diseases that are different. We know they're pretty different now in their origins, but a lot of times the clinical signs and symptoms are kind of similar. So I'll just stop there, and maybe if we will take a moment, and I'll have my guest, Dr. Karsten Baumann and Dr. Matt Harms, introduce themselves a little bit further and talk just a wee bit about what your background is and your research interest is in this field to start. So Dr. Baumann, if you wanted to start.
Yeah, hello. I'm Karsten Baumann. I'm a pediatric neurologist and neurogeneticist. So I got into the neuromuscular field really 30 years ago, almost 30 years ago, after finishing pediatric neurology. And I was interested in the genetic basis, genetic causation of neurological disease. And after my training completed in Boston, I was looking for the right lab to follow that. And it was Luke Kunkel's lab that took me in. And Luke Kunkel, of course, is known to probably most of you as the person who discovered, along with other people, the dystrophin gene, cause of Duchenne muscular dystrophy, which, at the time was a huge advancement in science. He identified this gene not knowing its function just by its genomic location. So that really started the era of genetic genomic medicine in neuromuscular disease. And so I joined his lab and got into neuromuscular that way and never left. And my research interest is really in early onset neuromuscular disease, pediatric neuromuscular disease, things that are often present when the baby is born with it. And more we are moving towards not only understanding the genetic basis of these disorders, but also thinking about the appropriate therapies that result from these findings. So, to give you an example, when I was in Lou's lab and he had discovered the dystrophin gene, and I was involved in finding some of the early LGMD genes in his lab, and now we're almost, this was the mid-90s, so we're kind of almost 30 years later, and both Duchenne muscular dystrophy as well as the LGMDs that I was working on, beta-sarcoglycan in particular, are now in gene therapy trials in patients actively being dosed. So that gives you an example or view how the field has advanced. For me personally, when I started out, really the focus was on finding those genes, and it took years to find a single gene. We're all proud to find a gene in a single disease, and that has now changed. Finding a gene or a mutation really brings with it the responsibility to deliver a treatment as well. So finding the genes and the mutations are now the beginning of a therapeutic journey for that. It's not the end of the journey, it's now the beginning of the journey. So that is really the paradigm shift that happened over the last years. For instance, we discovered a pediatric ALS gene just recently, SPTLC1, and with it, in the same paper, was a precision medicine approach to that disease. We discovered a rare mutation in a congenital muscular dystrophy that was hidden to us before because we couldn't look in the deep intronic parts of the genes. And finding that mutation inducing a pseudoexon delivered a precision treatment right with it. And so that is a paradigm shift that I've been experiencing as a neuromuscular geneticist.
That's great. Yeah. I mean, I know we have over 300 genetic causes of neuromuscular disease that we're tracking, and that number expands, it seems, weekly. Dr. Harms, do you want to talk a little bit about your background and interest in research?
Yeah, I'd be happy to. I'm Matthew Harms, and I'm a professor of neurology at Columbia University in the Institute of Genomic Medicine. So like Dr. Bonneman, I got interested in neuromuscular disease and uncovering the genetic basis. And like he just elaborated, really doing that as the beginning of a journey to then find some sort of a precision medicine approach that might allow us to zero in on a molecular therapy or a specific pathway or target in order to develop a drug or some sort of treatment that would undo or at least halt the neuromuscular weakness that was occurring. I started off by doing research uncovering the genetic explanations for limb-girdle muscular dystrophy and for spinal muscular atrophies. But then as the cost of sequencing got cheaper, realized that it was now amenable for more common neuromuscular disorders like ALS. And so I have, since 2015, been focusing much of my time uncovering new genetic explanations for ALS and trying to understand how it is that the genetic mutations that we do find then lead to neurodegeneration. And does that give us additional clues for pathways that should be targeted for therapeutic development? Sharon, to your point earlier about lumping patients together into neuromuscular disease because of the progressive weakness, I'm kind of a poster physician, if there is such a thing, for that, that lumping, because I see patients in the Pediatric Muscular Dystrophy Association clinic, the Adult Muscular Dystrophy Association clinics, and then the ALS center clinics as well. So I see kind of the whole spectrum of neuromuscular disorders and the whole genetic and lifespan of people with neuromuscular disease. So I think that I'm very comfortable with this grouping of patients. And I would kind of point out that in addition to just the historical manifestations that patients come, there are some very strong rationale for keeping this group of patients together. And that comes from the clinical trial outcomes assessments. So, for many human diseases, it's very straightforward. You're looking at a lab test that you want to reverse or an imaging finding that you want to prevent or see go the other direction. But for many of these neuromuscular disorders with progressive weakness as the outcome, you need carefully trained physical therapists and clinical trial outcome measurements that require a lot of training and are similar across those groups. So when it comes to kind of developing clinical trial infrastructure, I think there's also a strong rationale for many of these diseases kind of being held together in a group.
Yeah, no, that certainly makes sense to us. So at MDA, we have a patient registry called the Mover Patient Database, and we cover 7 different neuromuscular diseases in that registry. And it's everything from ALS, SMA, Pompe disease, which is a metabolic myopathy, to limb-girdle, the dystrophies, limb-girdle, muscular dystrophy, Becker muscular dystrophy, Duchenne muscular dystrophy. And although these diseases appear somewhat different in their cause, the case report forms for the data that we collected actually have a lot of common core data elements across all of these disorders. because they tend to have some similar effects, say on respiratory progression. A lot of them have cardiac manifestations that are similar. So certainly we see the commonalities there, and we have a lot of clinicians in our network who treat people in all of these different areas of neuromuscular disease. And so we touched just a little bit, I think both of you have mentioned the large number of genes that have been discovered in these largely genetic diseases. So when we think about where the field has come, that's one obvious difference, is we have a much better understanding of the genetic causes of the disease. Are there other things that jump out to you of the way that field has changed since you first entered the field, or things that you think are notable? I'll just throw that out to anyone who wants to jump in there.
Yeah, I would argue that it is not even just the genes that we're now discovering. It's actually individual mutations within genes that are coming to the forefront. And so I think we're moving post this a single gene, if you will, era into single mutation era, when we try to understand what they do mechanistically. So in the same gene, we can have different mutations causing completely different phenotypes. And also single mutations may have different levels of actionability, how you can address them on the molecular level. Some, as I mentioned before, for instance, with the pseudoexon, so a splice modulating therapy can be tailor-made to a particular given mutation within a gene. And the type of mutation then is more important than the gene, actually, and when you think about the treatments. So we are adjusting our molecular toolbox to what we find genetically, and that now drills down to the single mutation level. The other thing I think that is now moving into the forefront is the long realization that a single gene disorder is not a single gene disorder. Every single gene disorder is modified by genetic background, by additional genetic variants that play out in a single human being's genome. And that may be related to the primary disease, but it may also be related to the response to therapy. So for instance, is this therapy toxic for one patient and not for another? That probably depends on the genetic background of that patient. So the genetic complexity really goes down to single gene disorders as well as response to therapies. And then, of course, and Matt is much better suited to talk about that, there's a continuum from single genes to susceptibility, from single genetic causation to complex genetic causation. And ALS may be a prime example for that, to understand it. And I think what we're realizing in the rare disease field is, yes, that may be the case, but studying a single gene disorder in the sense that it is a primary gene that causes it with all the complexity coming behind it, as I mentioned before, really is informing us to also address more complex multi-genetic diseases. So I think the time spent in addressing single gene disorders is time well spent to understand more complex disorders as well. Matt.
No, that's a very good point. You know, that as we've developed the technology and the speed to be able to sequence, you know, large panels of potentially causative genes for ALS patients, you know, now in some places we're approaching comprehensive sequencing for entire ALS clinic populations. So like at the Columbia Clinic, every patient, you know, gets a broad panel looked at. And so we are appreciating individuals who have, you know, what people think of as kind of driver mutations, some of these monogenic explanations, which we now realize are not completely penetrant. So you can carry the gene mutation, but not everyone with the gene mutation goes on to develop the disease. And so can we look for and find those other modifiers that are leading to the expression of that gene in an individual patient with motor neuron degeneration? And, you know, by uncovering those modifiers, would we be able to impact the likelihood of developing that disease? We're also, you know, now getting to the point with close to 20,000 ALS genomes or exomes where we can start to look for patterns in what those modifiers might be, or look for those other kind of polygenic explanations for ALS, which I think will kind of lead to additional therapy ideas where you might be tackling multiple different pathways at once in a given patient based on the complex genetics that you were just alluding to. I was going to say the other kind of emerging driver of our ability to even talk about molecular, each mutation individually and understand each mutation individually, since they are so rare by themselves, is kind of the international collaboration and kind of the breakdown of silos between different research groups that are working on these things. So while, my project, Genomic Translation for ALS Care, has looked at 1,800 genomes, and Project Mine has looked at 6,000, and there are other groups that have looked at 2,000 or 3,000 here. The fact of the matter is that, one major change that's occurred since I entered the field is that now that data gets shared relatively quickly for overlapping information and for additional discovery that couldn't have happened when people were still, operating in their own research silos. And so that's one additional thing that I think has shifted over the course of the last decade that is really empowering kind of this next round of discoveries for neuromuscular disease.
Yes, and that actually it could be more extreme and say this is mandatory for us to move forward in the field of developing understanding and therapies for these disorders. And so it's so important for gene discovery and gene understanding, as you pointed out, but it's equally important for understanding therapies and developing therapies and therapeutic responses. Because every single rare genetic disease cause of, for instance, a form of ALS or other neuromuscular disease, may be small n's in terms of patients with that particular mutations, but every learning of a therapeutic approach in that patient group can inform a much broader group of patients and accelerate development of therapies down the line if the data is shared, if the data is made to be known. And that goes for the therapeutic approach used, the biomarkers used, the safety events that happened in it, the management of safety events, and the charting of outcome measures. I think every single rare disease, even small n's, can propel the entire field because every single one of these therapies makes broader points that are applicable across the field. And I think because of the commonalities that we have identified already in neuromuscular disease, neuromuscular is particularly powerful to benefit from that approach if we put it into action. And I think we're seeing a very, very promising moves that direction.
Yeah, so since I've been in the field, which was over 20 years ago when I started in the space, there are now almost 15 drugs approved for different neuromuscular diseases that are FDA approved drugs. So I think one huge shift I've seen is as we've understood the science better, it's been driving the technological development and driving everything that comes with it. So we've gone from a few companies in this space to several hundred companies who have things in the pipeline or drugs approved in the neuromuscular space. So that's created a lot of activity. Certainly has changed the environment quite a bit, mostly for good, I think. And another thing that I've noticed over time too is that the patient organizations in this space have become just more and more sophisticated. And the same thing that you mentioned about silos and collaborations we're seeing on the patient side as well. As these organizations mature, we've seen a lot more collaborative efforts, sharing of resources and information. And again, these groups, which are allied closely with the patient community and with the researchers, often are sort of the glue that pulls a lot of the different pieces of this research industry, different payers, even government pulls all of this together. It's sort of centered around patients themselves and patient advocacy organizations. So it feels like the field has matured quite a bit. We have, drugs approved. We have endpoints that have been used for drug approval, and that sort of paves the way for the next round of new therapies. And to use that as a segue, what is most exciting to you right now in the field? Maybe if you want to say a few words about what you're excited about seeing developing.
Yeah, let me start with some excitement and then also maybe with some challenges that the field is also experiencing at this point and has to face. The excitement for me, of course, as a neurogeneticist, is that we now have therapeutic options that move ever closer to the genetic causation of a disease. And I think if you think about genetic disease, it's a tough target to treat. It's a tough thing to treat, a progressive, often progressive disorder that affects such vital functions of the body. And really, your effect of your therapy needs to be as good as it possibly can be to really make a difference in the life of a patient. And the promise, of course, of gene-directed therapies is that we are moving closer and closer to the origin of the disease, and hence expect the impact to be greater. And that, of course, only works if we get there with our therapies, and we get there effectively. And when I say therapies, I mean the entire gamut of molecular therapies that are now on the table, from gene therapy, gene replacement therapy, to base editing, gene editing, transcript modulation. All of these are therapies that are gene and transcript directed, but they need to get there. And so this is one of the challenges that we face in the neuromuscular field. One is get to the target, get access to the target. So that's important in ALS, for instance, or disorders that are nerve-based, where you have to get to CNS origin of the disease with your therapies. And then on the muscle side, the pure size of the target, the size of muscle as a body organ is huge. And so we have to really, in that, cover the entire diversity of muscles that are important for movement, respiration, cardiac health, and life, essentially. And that brings with it distribution challenges and also safety challenges, of course, with the doses that have to be used to reach every single of these muscles. And so those are the target challenges. And the other challenge, of course, is the frequent progression that we see in many of our neuromuscular disorders. So we have to get there and treat effectively while there's still cells and tissue to rescue. So we have a window of opportunity that demands early treatment, early recognition, and early intervention.
Mm-hmm.
I think, Sharon, I would say that I'm most excited, and this is a little bit different between kind of the genetic muscle diseases and ALS, and that is in ALS, 5 to 10% of ALS patients have an easily identifiable driver gene mutation. And in some populations, that might even be up as high as 13 or 14%. But there's this large number of individuals that we don't yet have the ability to identify kind of a key monogenic explanation for their disease. But by studying, the 24 or so genes that have been implicated as causing ALS, we've been able to zero in on some key molecular pathways that could be targets for all of ALS. So when you study what's happening and going wrong in patients with C9orf72, you can see some downstream pathways that are disrupted that are shared with individuals with ALS that don't have an identifiable gene mutation in C9orf72. And so one of the things that I think has been very powerful in ALS that I'm excited about is that as we've looked at the implications of these driver mutations, we've zeroed in on particular pathways that are disrupted in essentially all of ALS and can go after those as targets. So I'm excited about molecular therapies that are targeting the autophagy pathways, or even the unfolded protein responses, the way cells deal with protein homeostasis and misfolded protein and aggregation, the way that RNA missplicing might be taking place when TDP43 function is not present in the cell nucleus, for example, and the way that that's being reflected in misspliced downstream targets that look amenable to potential therapies. And so that's kind of where I'm most excited, even as we don't have great driver mutation explanations for the vast majority of patients. And in fact, maybe we won't, but we've gotten really great insight into these shared molecular pathways within motor neurons and related cells that are key for the entire disease. So the kind of the springboard from the genetic explanations out into the rest of ALS is what I'm excited about.
Yeah, and that's a really powerful argument for identifying genetic causes and then understanding more sporadic disease. But also the complexity of genetic causation of ALS tells us that there probably is also complexity in sporadic ALS in terms of what an individual patient is a major driver amongst all of the downstream consequences. And I think the future, even here, will be precision medicine to understand this patient has this particular aspect of ALS pathogenesis as a major driver, and therefore we tailor make a treatment that addresses that predominantly, whereas another patient may have a different driver that is driving, and then we use a different therapy to address that driver, even in sporadic ALS. So I think the challenge here really is to continue to collect all of this data and all of the patients and really understand subcategories, understand individual drivers in all of these seemingly similar disorders.
Which raises the very important point of kind of figuring out biological markers of those different subtypes of ALS, right? And getting at, fluid and tissue biomarkers that might tell you, is this an ALS patient for whom neuroinflammation is the key driver, or for whom it's, purely a protein homeostasis challenge, to be able to kind of put patients into those different biologically similar groupings for clinical trials, for gene discovery, for mechanistic understanding, and what have you. And so, I think one of the other things that I'm very excited about in the field are all of the longitudinal biofluid biosample repositories that have sprung up, the natural history studies that are trying to link those biofluid changes to disease progression and disease manifestations. And even, one of the ways that we're going about that is by studying pre-symptomatic carriers of these gene mutations, sometimes even decades before we anticipate that they may develop symptoms, to see what are these biofluid changes, imaging changes that might herald symptom conversion, that might give us a handle on some of those earliest changes that take place, that we could then go and test in people who don't have genetic mutations to see if they're similarly occurring in advance of disease or at the time of symptom conversion.
And that gives you really the opportunity and hopefully a window into early treatment, perhaps even pre-symptomatic treatment, if your biomarkers are strong enough predicting a later manifestation of the disease. That would be, of course, wonderful if that materializes. But the other important, I think, of biomarkers that you mentioned and longitudinal following in these diseases, biochemically and clinically, is of course clinical trial design and clinical trial efficacy. So the more precise we are about the causation, the more precise we can be about the biomarker, and the more tightly we can design clinical trials that actually give us an answer whether an intervention is working or not. Because with so many causes, so many therapeutic avenues, and such limited resources, we need to be efficient in clinical trials and really doing what's necessary to tell us this is working and this is not working.
Yeah, you mentioned clinical trial efficiency. I think never has that been more important. As these many pharmaceutical companies are developing drugs for neuromuscular indications and wanting to bring them into clinical trials, is kind of the challenge on the clinician and clinical trial infrastructure that currently exists in most places. I think every center I know is looking to hire additional neuromuscularly trained clinicians to be able to help with the exciting clinical trials that are coming down the pipeline. We're all struggling to train enough clinical trial outcome measurement folks and get them as skilled as possible to be able to record clinical trials. And so I think as people are thinking about developing clinical drugs for clinical trials, we also need to be thinking about capacity within our neuromuscular centers to be able to execute on those clinical trials. And that's something that I know many centers are facing as a challenge right now, just needing well-trained research individuals to come on board and join us in this very exciting mission and at this very exciting time.
Yeah.
Great opportunity for trial networks and collaboration across sites. Mm-hmm.
Yeah, we're basically at a very exciting time where things are growing rapidly and our knowledge is increasing and we're making real progress. I think this is being felt by the patient community. But they're growing pains. And I think that's what we're putting our fingers on now, is the need to make sure that we are prepared to succeed by having all the resources that we need in place. And I think it's a good problem to have. It's definitely better, you know, than we were 20 years ago when we weren't sure what caused most of these diseases. So it's remarkable progress. It's a very exciting time. And I thank you both for this discussion, and hopefully you will enjoy the subsequent talks that will be focused on ALS. So thank you all.
Thanks, Sharon.
Thank you.